This is chromosomal basis of inheritance, chapter 24. So we first need to start talking about genetic linkages, and genetic linkages is just the way a group of genes is inherited. So all the genes on one chromosome form a linkage group because they tend to be inherited together. And as you can see here over on the right-hand side of the screen, I have an example listed. Gene 1, this is considered an unlinked gene. Um, there's genes located on different portions of the chromosomes, whereas genes 2, 3, and 4, these are linked together. So whenever they separate during meiosis and the sister chromatids begin to break apart, these genes are going to be inherited together. And as you can remember from the previous PowerPoint, whenever we started discussing physical characteristics such as eye shape, eye color, hair color, a lot of these are considered to be a linkage group, which means that they are inherited together. Um, so again, obviously this would violate the law of independent assortment. Because in the law of independent assortment, we learn that genes and alleles are inherited separately, that they do not depend on each other, whereas genes within a linkage group, technically those are inherited together because they're on the same gene. Um, If crossing over occurs, a dihybrid cross yields all possible phenotypes among offspring, but the expected ratio is greatly changed because the recombinant phenotypes are reduced in number. And that just makes sense because, again, we are not having independent assortment, which means that the possible phenotypes that could be displayed are going to be greatly reduced because, again, we have a linkage of these genes. So first, we need to start talking about X-linked inheritance. This is often called sex-linked inheritance, right? Because the X chromosome is going to primarily be for the female, and the Y is going to represent a male. So genetically female individuals will have two X chromosomes. There will be X and X, whereas a genetic male will have an X and a Y. Here we have an example of a sex-linked X-linked genetic disorder. So here we'll start out. The text says normally both males and females have 23 pairs of chromosomes. 22 of those are called autosomes, and autosomes, that is just a word that means that these are chromosomal pairs that are not the X or the Y chromosome. So the X and the Y chromosome are what's going to make you that's going to make up your genetic sex, or um, what characterizes you as male or female. So you have XY here, which is the uh, chromosomes for males. You have XX here, which is the chromosome type for females. So sex chromosomes, again, these are just XY, and we have 23 pair, so 22 of those are going to be what we call autosomes. So in X-linked inheritance, males have the sex chromosome X and Y, and females have two X chromosomes. Traits controlled by genes on the sex chromosome are said to be sex-linked. And that makes sense, right? We have sex chromosomes, and we have certain genes on these chromosomes which are whenever they are inherited through the X or Y chromosome, we will just call those sex-linked. That's where the lingo kind of comes from. A gene on an X chromosome is said to be X-linked, and a gene on the Y chromosome is said to be Y-linked. And for our purposes, we will not be dealing with any Y-linked inherited disorders because I'll just be quite frank, they're few and far between, whereas X-linked are going to be the primary modalities on which inheritance is saw. So for X-linked traits, a male always receives an X-link from his mother, and that makes sense. If you did a dihybrid cross and you saw along the vertical axis, you saw an X chromosome here and an X chromosome there, and if you had an X chromosome at the top with a Y, you're going to get your X chromosome from your mother, and that's very important because when we get to deal with these X-linked recessive traits or disorders, we're going to see that most of the time inheritance is passed from mother to son because again you get your X chromosome from your mother, and you inherit your Y from your father in the case of males. So the text says, because the X and Y chromosomes are not homologous, the Y chromosome from the father does not carry an allele for the trait. Most sex linked genetic disorders are recessive. Therefore, a female must receive two alleles, one from each parent, before she has the condition. And we can see that on this previous slide here. We have an unaffected father, an X and a Y, and we have a carrier mother, right? You can see here by the box that is not shaded in, she's most likely carrying this disorder, okay? She doesn't, again, this is a recessive trait, so in order for her to be displaying the phenotype, she would have to have a genotype that would have to be both little x's or um, both um, both of these boxes um, would have to be white. So here we see once they mate and they produce offspring that this child is going to get an X from his mother. He's going to inherit his Y from his father. And again, he's unaffected because he must have gotten this uh, this this blue box right here, which means that he is unaffected. And again, the daughter has to receive two of these alleles in order for her to be affected. So you would have to have an affected father for her to have a one of these white boxes to be displaying the phenotype. And she actually didn't get either. She also didn't get this. She's not a carrier either. We can see right here. And again, we have a carrier daughter here. As you can see, she got the normal X from her father, but she did inherit the defective X here. But still, she has to have two of those defects in order to be classified as being affected. Whereas the son only has to have one, again, because he has one X chromosome. Here we see that he receives the defective X chromosome from his mother, and of course he gets his Y from his father, so he is affected. So the probabilities, if you were if you're a mathematic person, you can see 50% one of two is affected, whereas 0% of the daughters are affected. And that would be approximately 50% of the daughters are carriers. Again, that would be one of two. We can kind of get into this. For X-linked traits, a male always receives an X-link from his mother, from whom he inherited an X chromosome. Because the X and Y chromosomes are not homologous, the Y chromosome from the father does not carry the allele for that trait. Most sex-linked genetic disorders are recessive. Therefore, a female must receive two alleles, one from each parent before she has the condition. And here we can see this displayed a little bit better, and we'll go through this table. And this table is primarily just talking about color blindness. And as I'm sure maybe some of you already know this, but red-green color blindness is an X-linked recessive trait, which means that males are going to be primarily more affected, again, because they only have to inherit one genetic defect, and usually that always comes from mother. Whereas females have to receive two. That means there has to be an affected father and there has to be a carrier mother. So we'll just kind of read the text here. The heterozygous female, the second genotype, so a big B and a little b, is sometimes referred to as a carrier female because although a female with this genotype has normal color vision, again, she doesn't have the two smaller lowercase b's um, as genotype 3 does. She is capable of passing on an allele for colorblindness. Females who are colorblind are rare because they must receive the alleles from both parents. And again, we've previously said that. That means she would have to have an affected father. She would have to have an affected father, one with a little b, and she would have to have a carrier mother. Males are colorblind. Males who are colorblind are more common because they need only one recessive allele to be colorblind. The allele for colorblindness has to be inherited from their mother because it is on the X chromosome. And again, I encourage you to do a dihybrid cross or I'm sorry, to do a cross of these, you can place an affected father, an XBY, on the horizontal axis, and you could place a carrier mother, an X uh, uppercase B and an X lowercase b, and you can see how um, the child who inherits the defective X allele, um, when I say defective, I just simply mean the lowercase b, that will be the male who is affected. Um, Now let us consider mating between a male with normal vision and a heterozygous woman. What is the chance this couple will have a daughter who is colorblind or a son who is colorblind? All daughters will have normal color vision because they all receive an XB from their father. Again, we said that makes sense. The sons, however, have a 50% chance of being colorblind, depending on whether they receive the XB or the XB from their mother. And again, we can kind of see that in this picture by the heterozygous phenotype. The inheritance of the Y chromosome from their father cannot offset the inheritance of an XB from their mother. Because the Y chromosome doesn't have an allele for the trait, it can possibly prevent colorblindness in a son. So again, here, this is the dihybrid cross that I was referring to. As you can see here, this is an X uppercase B and an X lowercase b, which means this is a carrier female, and then this is an unaffected father. So again, here, in the case of the two females, you will have one who is a carrier, and then you'll have one who is unaffected. But in the case of the male, you'll have um, a male who is unaffected, but you'll have a male who is also affected because, again, he inherited um, this defective allele or this recessive trait. So the text says the male parent is normal, but the female parent is a carrier. That is, an allele for colorblindness is located on the X chromosome. Therefore, each son has a 50% chance of being colorblind. The daughters will have normal color vision, but each one has a 50% chance of being a carrier. And again, here, this is just a pedigree, kind of going through this. I won't spend much time as I'll let you guys read this. Like colorblindness, most sex-linked disorders are carried on the X chromosome. The previous slide depicts a pedigree for an X-linked recessive disorder. More males than females have the disorder because recessive alleles on the X chromosome are always expressed in males. So again, whenever I would analyze pedigrees, whether it be in medical school or undergrad, one of the tricks that were a quick trick that was taught to us was to look at males. If males were primarily more affected, this was most likely an X-linked inherited disorder. As you can see on this previous slide, the grandson, he is affected. You have here a mother who is affected, and you have a normal father. So again, the child who is a male is going to inherit at least one of the recessive genotypes from the female. So some forms of color blindness are associated with recessive alleles on the X chromosome. The most common of these is red-green color blindness, and that's probably important for examination purposes. It would just be very easy to write a question and describe somebody who is unable to see reds and greens, but they can see yellows, blues, blacks, whites, and grays, and just want to know how this would be inherited. And again, that would just be X-linked inherited. I could describe to you somebody in this and say that their father is unaffected, and I would want to know what is the possible genotype of the mother. And again, that could be if she's not displaying the condition, and that would mean she's heterozygous, which means that she's a carrier. So her genotype would be X big B and X little b, meaning that the son who is affected most likely received that defective allele, being the recessive genotype. Duchenne's muscular dystrophy is an X-linked recessive disorder in which there's a mutation in the gene dystrophin. Dystrophin is an important protein that is needed for the muscle unit. Symptoms include a waddling gait, toe walking. They usually die by the age of 20 years old. Just want you to be able to recognize what Duchenne's muscular dystrophy is, know that it's inherited in an X-linked recessive disorder, and just be kind of familiar with some of the physical manifestations that occur. Usually the onset is between 3 and 5 years old. It's primarily males, again, because it's X-linked. Um, these are some of the signs that I won't test you on, but again, Gower sign is they are present primarily on their knees, and they push themselves up with their hands in order to rise. They actually have kind of larger calves, and that's called pseudohypertrophy because the muscle that's normally present within the calves is replaced by fat cells. Just kind of a tidbit for you. Fragile X is a syndrome that is due to an abnormal uh, number of repeat sequences. This is actually called a trinucleotide repeat. And Fragile X has some very unique manifestations, one of those being the long face and the prominent jawline. They also have genital abnormalities where they have macroorchidism, which is just large testes. They are very much on the autism spectrum disorder, and it is recognized as being one of the most common inherited mental impairments. Learning disabilities range from mild intellectual disabilities to severe. So again, just recognizing that this is an example of an X-linked disorder. Moving on is hemophilia, and again, philia means affinity for, and this is hemo means bleeding, so it's an affinity for bleeding. These patients inherit in an X-linked fashion. They're missing either, if it's hemophilia A, it's due to absence of factor 8, A for 8, or hemophilia B, which is just factor 9. These patients have a history of bleeding. Oftentimes they'll go and undergo a dental extraction or a root canal, and they have severe bleeding that takes forever for it to stop. And that's one of the questions they'll ask you at the dentist is, do you ever have a history of prolonged bleeding, or do you have hemophilia? And that's just something that dentists want to be aware of. If they are subject to minor trauma, such as external trauma, falling, they can have bleeding around the joints. So they can bleed externally from a cut, or they can have bleeding around the joints, and that can cause severe problems as well. So this is actually a very important slide that will be tested. This is called non-disjunction, and this is how we have a abnormal amount in the number of autosomes or chromosomes. So normally a human receives 22 pair of autosomes, like we previously said, and we said that autosomes were just chromosomes that were not X or Y. And do you remember what we said X or Y chromosomes were? Again, those are sex chromosomes. If you have two X sex chromosomes, then you are considered a female. If you have an X and a Y, you are considered a male. So there's actually a phenomenon called non-disjunction. And non-disjunction, the definition of it is failure of the homologous chromosomes to separate properly during meiosis. And as you can remember from the previous lecture, we talked about chromosomal separation. We talked about the law of independent assortment and the law of segregation, how the chromosomes will separate during meiosis. So what happens is you have a failure of separation of these homologous chromosomes. So what happens is you have, during fertilization, some of the, whether it happens in spermatogenesis, the formation of sperm, or during oogenesis, the formation of the oocyte, you will have a combination of a sperm who either has too many chromosomes with a female who has a normal, or you'll have a female with abnormal amount of chromosomes combining with a male who has normal. So this can result in either too many chromosomes or too little chromosomes. Here's a picture kind of depicting that, so I'll kind of read the text. Non-disjunction of chromosomes during oogenesis, so this is formation of the egg. Non-disjunction can occur during meiosis 1 and result in abnormal eggs that also have one or more less than the normal number of chromosomes. Again, the chromosomes aren't separating, so that kind of makes sense. Fertilization of these abnormal eggs with normal sperm can result in a zygote with abnormal chromosomes. So non-disjunction can also occur in meiosis 2 if the sister chromatids separate, but the resulting daughter chromosomes go into the same daughter cells. So again, fertilization of these abnormal eggs with normal sperm produces zygote with abnormal chromosome numbers. So essentially, this text is just wanting you to know this can occur in meiosis 1 or meiosis 2. So meiotic non-disjunction results in an abnormal production of either too many or too less number of chromosomes. Again, we should have 46 chromosomes, 23 pair. So you can have too little and result in 45. That's actually a syndrome called Turner syndrome, or you can have too many. There's several different types in which there's too many, and we'll kind of go over those. So this slide is kind of discussing the change in the number of autosomes. So normal development, we say that we have two of each kind. There are several different disorders in which you have trisomies, and again, that's just inheritance of three of a number of chromosomes. So again, there's going to be 23 different pairs. So there's a condition called Down syndrome in which you have, also known as trisomy 21, in which you have inheritance of an abnormal, um, um, in which you have inheritance of another 21 chromosome. So among autosomal trisomies, only trisomy 21 has a reasonable chance of survival after birth. There's also syndromes called Patau syndrome, and that should be chromosome 13, not 12. I apologize for that. I'll correct that. And then Edwards syndrome. And usually these children only live a few months after survival. Again, Down syndrome has a significantly different rate of survival than the average um, person with the correct amount of chromosomes. But again, that is going to be much better than these other trisomies, Patau and Edwards syndrome. So Down syndrome, some of the physical characteristics are going to include short stature. You'll see that as a common theme within all these trisomies. They also have flat faces, a palmar crease, mental impairment. I'm sure that you guys are well aware of that. It can be very mild or it can be severe. Uh, Almost all of these patients, if they live long enough, will develop dementia. There's also some cardiac abnormalities that occurs. Over here, I have a picture kind of depicting what this palmar crease is and this widened gap between the first and second toe. This is something as a pediatrician that you want to be well aware of. There are certain maternal blood tests that you can do prior to conception that will let you know if your child has a risk of Down syndrome. Um, and those are performed well in advance. But again, it's also important for the pediatrician to kind of do these inspections of the fingers and toes just to evaluate for any abnormalities. It's very important to know that I actually found this to be quite fascinating. Advanced maternal age is considered to be 35 because at the age of 35, the risk of Down syndrome, as well as other genetic defects, raises significantly. I can't remember the exact statistic, but if you look at the incidence in Down syndrome, once you reach the age of 35, it goes up significantly. Once you reach the age of 40, that is quite more drastic. This is called advanced maternal age, and you're actually considered to be um, a senile when you're giving birth at that age. I don't think that's really fair, but the risk is significantly more increased. And again, we don't really know why. There's ongoing studies trying to figure out what is causing this to happen. So again, Down syndrome, there are several methods which an individual can have Down syndrome, the most common being non-disjunction. Again, that's important, that word non-disjunction, which results in an extra copy of chromosome 21. Again, knowing chromosome 21 is important for examination purposes. This occurs usually during formation of the egg, during oogenesis. And again, I said previously, but again, here it is. The chance of a woman having a child with Down syndrome rapidly increases starting at about age 40, and the reasons are still being investigated. So you can also have trisomies in which it doesn't occur with autosomes, but instead it occurs with sex chromosomes. This can be either trisomies, and I don't necessarily wouldn't call it a trisomy, but polysomy would be the more correct term, in which you inherited either an extra X or an extra Y chromosome, or you can have the inheritance of too few chromosomes, and that would be one in particular that I'm thinking of known as Turner syndrome, and you'll see that on the next slide. But again, the text here says non-disjunction during oogenesis or spermatogenesis can result in gametes that have too few or too many X or Y chromosomes. If these gametes combine during fertilization, an abnormal number of sex chromosomes occur, leading to some of the conditions listed in the table. This is just a good table. Again, probably very high yield for examination purposes. I can probably bet my bottom dollar all these will be on here. Should be 3 easy points. Just flip to this slide and be able to tell me what these are. We'll quickly discuss these. Klinefelter syndrome is whenever you have inheritance of an extra X chromosome. Their genetic sex is going to be genetically male. They're going to have 47 total chromosomes. They have a very unique body stature in which they have a discrepancy in the waist to hip, or the torso to limb length. They're going to be very tall. They can develop abnormal breast development. They can often have breasts. The testes and the prostate is underdeveloped. They can also have difficulty with fertilization from a male perspective. So, sperm, because the testes aren't developed, the sperm may not be as have a high reproductive capacity. Again, they can range in terms of intellectual development. A lot of times they have difficulty with learning new things, difficulty with schoolwork, things of that sort. The sex chromosome combination is XXY, and that's significantly high yield. Jacob's syndrome is XYY. This is going to be a male, again, because we have two Y chromosomes. Anytime that you see a Y present in the sex chromosome combination, we're automatically going to call that male. The inheritance of an extra X does not really determine this. So this is going to be 47 chromosomes total in nature. Jacob's syndrome, these are going to be very tall males, again, much like Klinefelter syndrome. They're going to have persistent acne. Where this will probably differ is that they'll have normal behavioral development, whereas Klinefelter syndrome can display some type of learning difficulties. It was once thought, it was interesting, it was once thought that because of this inheritance of this extra Y chromosome, they were going to have more testosterone, which meant that they were more prone to undergo imprisonment or undergo incarceration. They were trying to do studies. We know that is absolutely not true today. So your book kind of makes the distinction that they develop normally during behavioral development. And I think that's an important distinction to make where this was previously thought. Turner syndrome, this is going to be genetically female. This is going to be inherited an XO. So they're going to have 45 chromosomes. Again, they're going to have an underdeveloped female reproductive system. If you are ever in medical school, you'll hear a term called streak ovaries. That's because their ovaries are very underdeveloped. Because their ovaries are underdeveloped, they do not undergo puberty or menstruation. Again, because they're not producing significant amount of eggs. The physical characteristics, they're going to be short stature in nature, broad chest. They call it a shield chest. That's also a buzzword for that. They can also have cardiac abnormalities too. Coarctation of the aorta, in which the aorta, the largest blood vessel in the body, has a narrowing. Usually they have to have heart surgery at some point. So again, Turner syndrome here, just kind of going over the physical characteristics. I've listed a slide over here for visual representation. They have small fingernails, web neck, poor breast development, short stature and broad chest. Just kind of seeing that web neck over here. Just want you to recognize that this is an example of a abnormal number of chromosomes where they have 45. And again, this is due to meiotic non-disjunction. Klinefelter syndrome, like we previously discussed, they have 47 chromosomes. There is a distinction where they say 48, but again, 47 is more common. This is going to be due to inheritance of an extra bar body, and a bar body is just heterochromatin, which is actually not active. So in males with Klinefelter, the testes and prostate gland are underdeveloped. Facial hair is usually lacking. They're usually slow to learn, but do not have intellectual disability. I apologize. They do not have intellectual disability, but they're slow to learn unless they inherit more than two X chromosomes. Again, here's a picture, kind of wanting you to display that. In terms of test questions, I'm not going to, this isn't a pathology class. Again, this is an introduction to biology class. Probably a question I would ask from this would be, can you recognize the genotype for Klinefelter syndrome? Can you recognize the genotype for Turner syndrome? I think that's more important than all the nitty gritty stuff. But I think it's important for you guys to learn kind of what you're dealing with, um, kind of what it looks like. Just be familiar with it. You don't have to know the ins and outs of it. Jacob syndrome, this is due to non-disjunction during spermatogenesis, in which you inherit an extra Y chromosome. Again, like we said previously, the males are usually taller than average, have persistent acne. Despite the extra Y chromosome, there's no difference in behavioral development. So, whenever you have chromosomal mutations, whether it be through different agents such as radiation from chemotherapy or certain organic chemicals, again, Roundup has been shown to cause DNA damage, or even various viruses. This can cause the chromosomes to break apart, and this causes chromosomal mutations in which you actually have breakdown of the DNA molecules within a chromosome. Again, if you guys previously remember from the last lecture, I discussed about the cell cycle checkpoints in which we detect DNA damage. That was P53. P53 was known to detect DNA damage, and you had tumor suppressor genes, which were supposed to stop abnormal oncogenesis. Um, so, if these cell cycle checkpoints are unable to regulate the cell cycle, you have DNA mutations accumulating. We know that DNA mutations, whenever they accumulate, they continuously undergo cell cycle division that leads to cancer or carcinogenesis. Um, Normally, whenever the breaks occur, the segments can reunite to give the same sequence of genes, but the failure of them to reunite can result in one of the several types of mutations. So there's deletions, duplications, translocations, and inversions. Just want you to know the definition of each. I've kind of included some examples, but more important for you guys is just to know the definitions. A deletion, a chromosomal deletion, occurs whenever you have a single break in a chromosome and it causes us to lose an end piece. Over here on this picture, I have a depiction of chromosomal deletion. As you can see here, this chromosome is significantly larger than this, and that's just because we've had breakage of one of the chromosomes. An individual who inherits a normal chromosome from one parent and a chromosome with a deletion will obviously be lacking the alleles from the inherited chromosome that had the deletion. And as an example, a syndrome can occur. This happens with a syndrome called Williams syndrome. I very much encourage you to Google this one. The children have a unique phenotype. They're very friendly in Williams syndrome, characteristically, but your book describes them as looking like pixies. They have turned up nose, they have wide mouth, small chins, and large ears. Chromosomal duplication, again, this kind of makes sense. If you think about it, you're just duplicating this chromosomal segment right here. The individual has more than two alleles for certain traits. We talk about here an inverted duplication called inversion dup 15 syndrome. These children have poor muscle tone, mental impairment, seizures, a curved spine, and autistic characteristics. So this goes to show you that sometimes too much of something can sometimes be a bad thing, and that occurs kind of with these duplications. So again, just knowing the definition, that's primarily the most important. Dup 15 is just included as an example. Translocation, again, location, translocation meaning movement. So movement of a chromosome segment from one chromosome to another. The translocation can either be balanced or unbalanced. Whenever we have balanced translocations, the individual is unaffected because they can receive one normal copy of the chromosome from the normal parent and one of the abnormal chromosomes. So if it's unbalanced, meaning that there's extra material from one chromosome and the missing material from another chromosome, so these children can often result in miscarriages. So some of the causes of miscarriages largely are due to unbalanced translocations of chromosomes. Essentially, it's just going to produce life that is not viable. It's not capable of living. But if the children do live, they can have severe symptoms as well. An inversion occurs when a segment of a chromosome is turned 180 degrees. Your book makes this distinction, and I think this is very good. If you were to ask someone not in the scientific community if you thought this would be a problem, they would probably answer no. But because you're reversing the sequence of alleles, this can lead to altered gene activity. So crossing over between an inverted chromosome and the non-inverted homolog can lead to recombinant chromosomes that have duplicated and deleted segments. Thank you so much.